All right, so next up, we're going to talk about proteins. This is a large class of very important biomolecules. We are going to talk about the 4 different levels of protein structure, and then we're going to talk about how the structure of that protein affects its 3-dimensional shape. And because you guys are catching on by now, we know that the form, the 3-dimensional shape of a protein is going to relate to the job that it does, its function. Form follows function. All right, so what do proteins do in the body? All the dang things. They can be structural, so are your collagen that makes your skin elastic. They can be for regulation, like moving things around in the cell when it's time for the cells to reproduce. They can be contractive, so that can allow your muscles to contract so that you can move around. They can be protective, like the keratin in your fingernails. They can be used for transport, like the hemoglobin that moves oxygen and carbon dioxide around your body. They can be used as storage. The protein in milk, casein, is broken down to release energy. We're going to have a whole chapter about all sorts of proteins stuck in the cell's plasma membrane that allow things to move in and out of the cell. They can be toxins. And really, what we're going to spend a lot of time talking about in this course is the function of enzymes, because enzymes speed up how fast a chemical reaction goes, allowing life to continue. All right, so let's look at some other hormones. These can be small proteins. Sometimes they're steroids, which is a lipid. We'll talk about that. That can regulate or control how things respond. So, for example, insulin is a mechanism. It's a small protein that's made in the body that will bind to these other proteins that are embedded in the cell's membrane that will allow glucose to enter the body, to enter the cell so that we can use it for cellular respiration. So the reason that I bring this up is that my kid over here, Sammy, was diagnosed with type 1 diabetes. In 2023, question mark, about then. And which means that their body no longer produces the protein insulin, and therefore they have to take it externally. Okay, we talked a little bit about enzymes, but here we are again. Oh, look, this is Sammy again. This is little Sammy when she was just a wee lad. And it's a Sadie. Cute little naughty dog. Okay. So in order for a chemical reaction to occur, we have to add some energy to destabilize our starting molecule so that we can get our ending molecule. Now, what an enzyme does is it lowers that amount of extra energy that we have to put in to that system. So without, change colors. Without the enzyme, we have to add this much energy. With the enzyme, we only have to add that much energy. So that's going to make our speed up our chemical reactions. The reason that I have my dog Sadie and my kid Sammy on this is because if you've ever been a parent or a pet parent and you hear something happening in the other room, but oh dang, you just sat down, but you know that if you don't go see what those guys are getting into, it's going to cost you a lot of energy later. So, you have to harness that extra energy to get up and go prevent them from digging in the garden or for helping to make bread. All right. So, our monomers of proteins are called amino acids. Okay. There are 20 different kinds of amino acids that are biologically important. 11 of them humans can make. We can produce them in our own bodies. But the other 9, humans can't make. So we have to get them from our diets. We have to consume them. So those are called our 9 essential amino acids, if you've ever heard of things. Okay, let's look at the chemical structure of amino acid real quick. So this carbon is bound to 2 oxygens. This guy has a little hydrogen over here. This is called our carboxyl group. Okay, that's going to be important because I'll get there eventually. Okay, in our amino group, we have a nitrogen, and that is has a couple hydrogens on there. Okay, all right, so let's look at this. Look at this carbon. Here we just got our little placeholder hydrogens. Anytime we don't know what to do with an extra bond, just throw hydrogen on it. Okay, but then this has this thing called the R group, or the R side chain, and the R group varies to produce those 20 different amino acids. Okay, so we're going to look at the structures of these R groups. Okay, but you can kind of think of this like the X in algebra. Okay, the X in algebra can stand for anything. Okay, the R in our R group can be any important type of side chain. So let's see what we got going on. Okay, so here is all of the amino acids that are biologically important. Now, please do not freak out because you do not have to memorize the structure of these. I did when I was in biochemistry as an undergrad. I memorized the names, the codes, and the chemical structures. I took the exam and immediately info dumped all that stuff. Okay, so what we're going to focus on for a minute is looking just at the similarities and the differences between these. So let's start with tyrosine. Okay, here's our amino group. Here's our carboxyl group. There's our little carbon hiding in there. There's our little hydrogen hiding in here. And then this is our R group. Okay. Let's look at, hold on, I have to get a chair because I'm short. Okay. Let's look at alanine because he's just a wee little lad. Okay, here's our amino group. Here's our carboxyl. Here's our R group. It's just a carbon with 3 hydrogens on it. Okay. Now we remember. Hopefully we remember. Are the bonds between our carbon and our hydrogen, are they polar or nonpolar? These are nonpolar bonds. So our friend alanine is going to be nonpolar. It's going to be hydrophobic. Okay. Let's scooch over and look at our boy serine. Here's our amino group. Here's our carboxyl group. Here's our R group. We have a carbon. I'm going to just expand this out a little bit for you. Okay, so that's what this CH2, that's here. Our OH is here. We know that this bond is a polar bond. And so therefore, it's going to be hydro. Spelling. No, I did it right. Hydrophilic. where it loves water. Okay. Let's look at arginine. Amino group, carboxyl group, R group. Aha, look at here. We have a charged particle. Okay, it can be a positive charge. This dude over here is a negative charge. And when we see a positive or a negative charge, we know that that thing is going to be hydrophilic. Loves water. Wants to hang out with water. So I will just do a NH. Double bond over here. We know that these are polar. So let's just remember that our partial negative charge of our water molecule is going to be forming hydrogen bonds, boop, 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 with that hydrogen. Okay, this is also positively charged. So our negative sides of our water molecules are negative. They're going to be hanging out with that positively charged nitrogen. Okay, so the R groups are going to be what gives our amino acids their chemical properties. Okay, whether they're acidic or basic, whether they're polar or nonpolar, whether they're hydrophilic or hydrophobic. Now, as you go on in your careers, you might get to a level where you need to memorize the chemical structures of these. But for this class, for now, if I need you to know that a particular amino acid is either hydrophilic or hydrophobic, or acidic or basic, I will tell you that information. You don't have to memorize all 20 of these and all of their chemical properties. Okay, I will give you that information if I need to ask you a question about that. Polymers for proteins are called polypeptides. Okay, poly, we know that means many. And then whenever you hear peptides, you're going to be like, oh yeah, that mofo, that's a protein. Okay. We call them polypeptides because the bond that holds the two amino acids together is called a peptide bond. It's a particular kind of covalent bond. Between two amino acids, and I'm going to write amino acids AA because I'm lazy. Okay, so here again, this is our amino group. This is our carboxyl group. We're going to have a dehydration reaction where this oxygen and these two hydrogens go away, whoop, form our water molecule. And now this carboxyl group of that amino acid is linked to amino group of this amino acid. Okay. So then we could throw another amino acid here, another amino acid here, blah, 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 blah. Okay, however many amino acids long our polypeptide is going to be. All right, so let's talk about these four levels of protein structure. Okay. So what we're seeing here, this is modeled forms that this protein called Rubisco can take. Rubisco is what is going to allow plants to do photosynthesis. That's what's going to grab the carbon dioxide from the atmosphere, boom, and then start it on its path to become glucose. Okay. But the reason that I bring it here is because there's actually a whole bunch of different polypeptides that are required to get together in order for this protein to do its job. Okay, so that'll come back in. Okay, all right, so the first structure is called the primary structure. And if you want a little shorthand, you can just do it right in number one with like that little degree symbol, and that can mean primary. Okay, so this is the first structure. So this is just the order of amino acids and how they are linked together in that chain, right? So first we're going to have glycine and then isoleucine and then valine and then blah, 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 blah. Okay, just what order are we putting those amino acids in? Okay, when we link them up in that peptide bond. So here's a dude called Fred Sanger. He's a super cool guy. He has 2 Nobel Prizes. He's one of 4 people. There's only 4 people on the planet that have earned 2 Nobel Prizes ever in history. And our boy Freddie is one of those. Okay. He developed a way that we can take a protein and figure out the primary structure or the order of the amino acids. In fact, insulin was the first protein to be sequenced using Fred Sanger's way. And a friend of mine is now using a derivative of Fred Sanger's method that he developed in the 60s to be screening young children for the probability of developing type 1 diabetes later in their life. So they can take a little blood sample, take all those proteins, sequence them, and then see if there's any proteins that are found more often in individuals that develop type 1 diabetes later in life. So just a cool application of what's going on here. Okay, so the primary structure or the order of the amino acids in a protein is determined by your genetic code, so your DNA. Okay, and we're going to spend like 3 chapters talking about how we actually go from being a sequence of DNA to being a protein. Okay, but if we have changes in the genetic code, that could result in a change of the primary structure. So we'll use hemoglobin as an example. Okay, so if you've ever heard of sickle cell disease, that is 1 amino acid change. So here's the amino acid sequence of a normal hemoglobin, right? We're really just looking at 3. So we have this threonine, we have proline, and then we have glutamic acid. Oh, and then there's a little hiding lysine in here. Okay. Now, people that have sickle cell have a genetic mutation. So instead of calling for glutamic acid, which is, there's our OH group, this is hydrophilic. What we actually end up with is, that's the same, that's the same. Uh-oh, now we've got this thing called valine. And then we got lysine. Oops, let me not circle that. And then we got lysine again. Okay. So if we look at the chemical structure of valine, oh no, we're going to see that that amino acid is actually hydrophobic. And so that's going to change the way, so this little amino acid that's hiding in here, let's see, one of the things over here, is going to change the structure, the 3-dimensional shape of that protein is changed. And so therefore, we don't have this little donut shape anymore for oxygen to fit in. Where do we put the oxygen over here? There's no place for it. Okay, so talked about the primary structure. That's just the order of amino acids. Next is our secondary structure. Again, if you want a shorthand, I just do a 2 with a little degree mark. That means secondary. And this is going to, the interactions between the amino acids, whether they're hydrophilic or hydrophobic, whether they can form these hydrogen bonds, that's either going to get them into this thing called a beta sheet, where it's pretty flat, or we can call it an alpha helix or an alpha coil, where they're going to kind of be twisty. So this is the two-dimensional shape that a protein takes, right? So it's the folding of the coiling in two-dimensional space. Okay, and then this, again, this is a midway point in forming that eventual functional structure that we're all going for. All right, so next is our tertiary structure. Okay, tertiary just means it's the third one. Okay, our tertiary structure, this is our complex three-dimensional shape. This three-dimensional shape is due to those chemical interactions of the primary structure. Remember, those are our amino acids sequence. So positive and negative charged amino acids are going to be attracted together to each other to form this ionic bond. We see that here. We can have hydrophilic, hydrophobic interactions. So here we can see some hydrogen bonding going on over here. These guys are hydrophobic, so they're going to be hiding in the middle, away from where the water is. Some of our amino acids have sulfur in them, so the sulfurs like to link together to create a disulfide linkage. And if you're familiar with the sticky smell of skunks, that's due to a disulfide chemical structure. People that have curly hair also have more disulfide links to have the keratin of their hair be curly. I don't have that. My hair straight AF. All right, and our last but not least is where we have our quaternary structure. And then I always make the joke that this is the four-dimensional shape. No, that's not true. Okay, that's just a joke. Okay, but we're going to have all of these multiple polypeptides, right? They each have their own three-dimensional shape, and they're going to come together as the various subunits to create a functional protein. Okay, so here's a polypeptide. This is another polypeptide. So on their own, polypeptide 1 and polypeptide 2 don't do a formal job, but when we link them together, then they can function. So remember that enzyme Rubisco and hemoglobin both have multiple subunits, 4 or more subunits that are important to their structure and function. Okay. All right, so we have already talked about the genetic mutations that we have in DNA can change the shape of a protein and therefore change its job because form follows function. Okay, this is particularly especially true for our proteins. Okay, so changes can come because of genetic mutations, so errors in the genetic code, or we can denature a protein by permanently changing its three-dimensional shape due to environmental changes such as pH, temperature, chemicals. So if you've ever had a fried egg, you know that when you apply heat to that, it changes the chemical structure of the egg white and the egg yolk, and changes it by denaturing. So it's no longer going to do the job that it would do for a baby chicken, but it's going to be a yummy breakfast for you. Okay, when we talk about enzymes later in the semester, we're going to talk a lot about this denaturing and the importance that it has.